Now let's look at how DNA replicates itself. One of the most significant features of the double helix model was that it suggested the basic mechanism of how DNA is replicated. Three alternate hypotheses were proposed on how the old and new DNA strands interacted during replication. In the conservative replication model, the parent strands act as a template for the synthesis of an entirely new DNA molecule. The two parent strands reassociate after acting as a template, thus restoring the parental double helix. As you can see in the figure, after the second round of replication, the parent DNA strand is still visible in dark blue, and three brand new DNA molecules are produced in light blue. In the semi-conservative replication model, the two strands of the parent molecule separate and each strand functions as a template for the synthesis of a new complementary strand. As a result, the newly produced DNA molecule will have one old and one new strand. In the dispersive replication model, the parent DNA molecule is cut into sections so that the newly produced daughter DNA molecules will contain a mixture of old and newly synthesized DNA. To determine which of the three hypotheses of replication is correct, Mieselson and Stahl designed an experiment that distinguishes between the three models. For this experiment, they grew E. coli bacteria in the presence of heavy nitrogen to label the bacterial DNA called generation 0. They then moved the bacteria to a medium containing lighter nitrogen to label the newly formed DNA strands as the bacteria were dividing. Samples were collected during generations 1 and 2. The DNA from the bacterial samples collected during each generation was extracted and centrifuged to separate the DNA based on density. In generation 0, the DNA was labelled with heavy nitrogen and therefore appeared at the bottom. Generation 1 produced an intermediate band between heavy and lighter nitrogen. Generation 2 produced two bands, one at lighter nitrogen suggesting newly formed DNA and one intermediate band suggesting a hybrid. These results supported the semi-conservative model of DNA replication, where the parent strands serve as a template for the new daughter strands. Now let's reflect on the learning objectives we've covered so far. You should now be able to describe the semi-conservative model of replication and outline the experiment that confirmed this model as the correct model of DNA replication. The enzyme that catalyzes DNA synthesis by adding nucleotide bases to the new strand is called DNA polymerase. DNA polymerase is an enzyme, and it can only work in one direction, adding nucleotides to the 3' end of the growing DNA strand. As a result, DNA synthesis always proceeds in the 5' to 3' direction. In this image, thymine is added to the 3' end of the growing DNA strand, being complementary to adenine at the parent strand. DNA polymerase catalyzes the addition of nucleotides like this by releasing two phosphates. This way, DNA polymerase adds nucleotides in the 5' to 3' direction, and the daughter molecule grows as indicated by the red arrow. Replication occurs during the S phase of the cell cycle. It begins at special sites called origins of replication. In bacteria, this site is a specific sequence of nucleotides that is recognized by the replication enzymes. These enzymes separate the strands, forming a replication bubble. Replication proceeds in both directions until the entire molecule is copied. In eukaryotes, there may be hundreds or thousands of origin sites per chromosome. Once started, replication proceeds in both directions from each starting point. Multiple replication bubbles form and eventually fuse, thus speeding up the copying of very long DNA molecules. DNA synthesis involves a sequence of well-organised enzymes that are joined into a large multi-enzyme complex called the replisome. For DNA to replicate, the double helix structure must be opened. Helicase is an enzyme that unwinds the double helix at the replication fork and breaks down the hydrogen bonds between complementary bases to separate the parent strands. Single-stranded binding proteins attach to the separated strand and prevent them repairing. The unwinding of the double helix causes tension further down the helix ahead of the replication fork. Topoisomerase helps relieve this tension by cutting and then rejoining DNA. DNA polymerase can't initiate the synthesis without the presence of a primer, which provides a free hydroxyl group at the 3' end so that DNA polymerase can add incoming nucleotide bases. A primer is an RNA strand with a few nucleotides that are complementary to a small region on the template strand, and it's synthesized by primase. DNA polymerase III adds nucleotides to the free hydroxyl group at the 3' end, and the leading strand or continuous strand that replicates towards the replication fork is produced. The other DNA strand that's synthesized away from the replication fork is called the lagging strand. This occurs because DNA synthesis must proceed in the 5' to 3' direction. The lagging strand also starts when primase synthesizes a primer for DNA polymerase III to add free nucleotide bases to the 3' end of the primer. In contrast to the leading strand, the lagging strand requires multiple primers that are synthesized separately. The short segments of the lagging strand that are synthesized by DNA polymerase away from the replication fork are called Okazaki fragments. DNA polymerase I is an enzyme that removes the RNA primers from the lagging strand at the start of each Okazaki fragment and fills the gap by adding nucleotides. The enzyme ligase then joins multiple Okazaki fragments to form a continuous DNA strand. Take your time to review the roles of the various molecules that play a role in DNA replication so that you're familiar with them. Now let's reflect on the learning objectives we've covered so far. You should now be able to describe the process of DNA replication, including the role of the replication fork and the origin of replication. You should be able to explain the roles of DNA polymerases, ligase, primers, primase, helicase, topoisomerase, and single-stranded binding proteins. You should be able to describe the anti-parallel nature of DNA and explain why continuous synthesis of both DNA strands is not possible. Distinguish between the leading and lagging strands. Explain how the lagging strand is synthesized even though DNA polymerase can add nucleotides only to the 3' end. And describe the significance of Okazaki fragments. The DNA polymerases are highly selective in matching complementary bases correctly to the growing DNA molecule. Incorrect nucleotides are inserted at a rate of about 1 per 100,000 bases. Fortunately, DNA polymerase can proofread its work. As soon as a nucleotide is attached to the growing DNA strand, DNA polymerase checks the match between the paired bases. And if a mismatch is detected, it can correct it by replacing the incorrect nucleotide. DNA polymerase can do this because it can act as an exonuclease that removes nucleotide bases from DNA. Mismatched nucleotides can sometimes evade the proofreading process. Repair enzymes can remove the defective bases and repair the DNA strand in a process called mismatch repair. This process depends on the template or parent strand to correct the mismatch. DNA can also be damaged by exposure to harmful chemicals or physical signals such as cigarette smoking and X-rays or UV light from the sun, along with spontaneous changes from cellular metabolic products such as reactive oxygen species. Two processes repair damage in a single strand of DNA, where an enzyme called a nuclease cuts out the damaged stretches of DNA and the gap is filled with the correct nucleotides by DNA polymerase, and ligase seals that gap. A single incorrect or damaged nucleotide base is repaired by base excision repair. Nucleotide excision repair removes helix distorting lesions such as pyrimidine dimers that are caused by exposure to UV rays such as the light from the sun. The regions at the end of eukaryote linear chromosomes have telomeres, which are short repeating stretches of nucleotide bases. Whenever DNA is replicated, the chromosomes tend to become shorter. That's because the RNA primer at the end of the lagging strand can't be replaced with DNA because there is no 3' end available for DNA polymerase to add free nucleotides. As a result, a section of single-stranded DNA in the lagging strand is left unpaired at one end of each chromosome. This remaining single strand of DNA is eventually degraded, shortening the chromosome length. The length of chromosomes plays a key role in determining how many times a cell can divide, or the life of a cell. Telomerase is an enzyme that can repair the end of lagging strands by adding more bases. Telomerase is inactive in almost all cells except for embryonic cells and cancer cells. Now let's refresh on how our cells are able to fit 2 meters of DNA within their nuclei. Eukaryote chromosomes have linear DNA molecules and they're associated with a large amount of protein, mainly histone proteins. The histones bind to each other and to DNA to form nucleosomes, the most basic level of DNA packing. We call this DNA and protein complex chromatin. During interphase, much of the chromatin is compacted into 30 nanometer fibres through folding and looping. Here we can see how looping of this fibre forms domains. During metaphase, these domains then coil and fold to produce a compact metaphase chromosome. Interphase chromosomes occupy restricted regions in the nucleus, as we can see in this micrograph of a cell. The loosely packed chromatin is called euchromatin, while the highly condensed chromatin, where genes aren't expressed, is called heterochromatin. Let's reflect on the learning objectives we've covered so far. You should now be able to explain how mistakes are repaired in DNA replication, and after you've looked at the case study in the tutorial, describe a disease or condition that arises as a result of errors in these pathways. Understand why DNA replication in linear chromosomes can lead to shortening and how this is overcome in some cells, such as embryonic cells and cancer cells. And you should be able to describe the nature of DNA packing and understand the roles of nucleosomes and histones. Thank you for watching.